right, so now you need this diagram. You also need your same sheet of paper that we just completed with the first diagram, as well as colored pencils. So now we're just going to look at the muscle fiber itself. Right, so make sure that this is labeled subcellular structures of the muscle fiber. Right, so we're just looking at the muscle cell itself. And we've already labeled these before on your legend, so we just need to add the numbers now. Right, so number 9 is the muscle fiber. Right, we already have this on your legend, so just draw a circle, right, put a little number 9 there. Right, so this is the muscle fiber, which is the muscle cell. And each muscle cell, just like any cell, has a cell membrane, has a nucleus, and in this case we have multiple nuclei, and then cytoplasm. Remember, we're rebranding the cell membrane. We can't call it a cell membrane because this is a muscle fiber. So we call it the sarcolemma. So here, this entire structure, right, that's our cell membrane or the sarcolemma. So this is 9A. Then we have in green here and then here, we have the nuclei, right? So here's one nucleus, here's another nucleus. Then with 9C, this is going to be the sarcoplasm. Don't color too deeply here, right? Just kind of, you get the idea that it's cytoplasm. Then I'm going to show you the sarcoplasmic reticulum. Remember, the sarcoplasmic reticulum is just a network of membranes that stores and releases calcium. So with this sarcoplasmic reticulum, it has various pieces and parts, and this is new now, right? So the CSR, right? The CSR, this is just called the cisternae of the sarcoplasmic reticulum. Cisternae just refer to enlarged areas. So it's still part of the sarcoplasmic reticulum, but notice that this tube and this tube is much larger in diameter compared to these other tubes. So it's an enlarged area or a widening, if you will. And these are in direct contact with something called T-tubules, right? T-tubules. T-tubules stands for transverse tubules. All right, so again, it's all about the neighbors. So here we have the CSR, which is the cisternae of the sarcoplasmic reticulum. And then we have T-tubules in between. So we'll color those in just a second. So in orange here, right, in between the cisternae of the sarcoplasmic reticulum, we just write TT with an arrow. TT stands for transverse tubules. And these are just invaginations of the sarcolemma. With an invagination, just picture pantyhose, right? And if you take pantyhose and you stretch them out, and then you push your finger into the pantyhose themselves, right, on the outside. Oh, I don't like where this is going. Let's do an example. Okay, if you take a cell membrane, you push your finger into the cell membrane, right, and some of that cell membrane is going to be taken, right, and pushed deeper into the actual cell itself, right? That's an invagination. So these are invaginations of the sarcolemma. So they pass deep into the substance of the muscle fiber, right? Meaning that parts of the cell membrane, right, the sarcolemma, are actually being carried deep into the muscle fiber, right? So if this is a round tube, right, we're going deeper into the cell itself. And then when we have three structures together, right, just this little bracket here, just write TRI. The TRI stands for a triad. It's between the cisternae of the sarcoplasmic reticulum, so in blue here, plus the transverse tubule, plus another cisternae of the sarcoplasmic reticulum. So these are areas where the transverse tubules meet the sarcoplasmic reticulum. So 3 structures, triad. Then in red, this is number 10. And again, we already have number 10 labeled, right? These are myofibrils, which are made up of myofilaments. And this was already on your key, right? So just label number 10 here. Now we move to the next diagram. It's actually in the same sheet, but it's just that smaller picture. And this is just a zoomed in version of the myofilaments. Right, so we went from myofibrils, and myofibrils are made up of myofilaments. So what you're seeing is just a zoomed in version of the myofilaments. So adding onto your sheet, right, in blue here, number 12. Number 12 is going to be the thick filaments, which are just bundles of myosin molecules. So these are the thick filaments. Notice all together, right? This is a thick area, so they're myofilaments, so they cleverly named them thick filaments. And if it's bundles of myosin molecules, right? Myosin, think of it as being 2-headed golf clubs, right? And so here, there's just one golf club, right? 2 heads. 12A, this is just our myosin. Myosin is just a contractile protein. Then with 12B, we're just going to split that myosin now into different structures. So we have a myosin tail, or the body. And this is just the horizontal portion, and it acts as an anchor. Basically, it doesn't do anything. It just lays there, right? And just anchors the myosin to other myosin molecules. 12C, right, which I have circled here, we have the myosin heads. There's 2 heads. One contains an actin binding site. An actin binding site, it tells you what it's going to bind to. Actin. The other one contains an ATPase site. So ACE indicates enzyme. That's going to break apart ATP, right? ATPase site. So an actin binding site, because it binds to actin, and an ATPase site. 12D, we're just going to call this the hinge between the head and the tail. Right? So this little hinge, it swings, right? It's really just picture a sperm doing a sit-up, right? So there's a hinge, right? And here, right, we see its tail, here we see the heads, right? And so it's a sperm doing a sit-up, right? So here we have the hinge between the head and the tail. Moving on to 13, right? These structures out here, these are thinner. So wait for it. They're going to be called thin filaments. And these thin filaments are comprised of actin, tropomyosin, and troponin. So we're going to split the thin filaments at the molecular level. So 13A, right, it looks like pearls, right, or a beaded necklace that's been twisted. This is going to be our actin. Actin is a contractile protein, and it has myosin binding sites. If the name is myosin binding sites, it's going to bind to myosin. Remember, actin and myosin were the contractile proteins, but our molecular chaperones are going to be tropomyosin and troponin. So 13B, this is our tropomyosin. And it's just a long filament. And basically its job is just to block things, right? If you laid down in the middle of the classroom you would just be blocking things, right? So it's a filamentous blocking protein, right? And what it's doing is it's blocking these myosin binding sites, right? So it's a chaperone, right? Think of any school dance when you were like in 6th grade. You had a bunch of chaperones, right? Preventing everybody from coupling, right? No touching. So tropomyosin blocks the myosin binding sites on actin. And then in blue here, 13C, we have troponin. Troponin, I like to call the molecular staple. Right? It's going to hold tropomyosin on, right? Because tropomyosin's just lazy, right? It's just laying there. It's just blocking everything. And so troponin actually does all the work. So troponin holds tropomyosin in place, and it has a binding site for calcium. Cleverly, that is called, for those of you who are curious, the calcium binding site. So when troponin binds to calcium, it undergoes a conformational change. Conformational just means shape change. And because troponin is holding on to tropomyosin, if troponin undergoes a shape change, it pulls tropomyosin with it. Right? Tropomyosin, again, it's just lazy. It's just laying there. But if troponin moves, it's hanging on to tropomyosin, it's just like moving a really thick rope out of the way.